Thank you, Nesar, for providing this opportunity to present my work. So, my title is Frailty-Based Plasma Proteomic Signature Capturing Overall Health and Well-Being. So, frailty is defined as biological syndrome of decreased reserve and resistance to stressor, resulting from cumulative decline across multiple physiological systems. And it causes vulnerability to adverse outcome like mortality, hospitalization, falls, etc. So this is an example, like if there is a minor illness in a fit older adult, the person comes back to normal after that illness get over. But when the person moved towards the frailer state, it moved towards down, like functional abilities comes down. How to quantify frailty? There are basically two main strategies which is used. One is the physical frailty definition, which is based upon 5 components, including unintended weight loss, self-reported exhaustion, weakness, which is mainly the grip strength, then slow walking speed and low physical activity. But our main focus is on the cumulative deficit frailty index, where frailty index assesses the frailty as an accumulation of deficit over a wide range of health outcome. If you look there, there is disability where all the ADLs go into the scale, then cumulative medical condition, then all the cognitive tests are included, as well as even the nutrition. So it's basically a easy construct. Frailty index is number of deficit in an individual divided by total number of deficit measured. So if there is like a 10 deficit in an individual out of 50 health deficit measures, the frailty score of that individual is 10 by 50, coming to 0.2. Then we have an in-house cohort at Einstein, that is longevity cohort, which is consists of community residing Ashkenazi Jewish seniors. So this study was mainly planned for understanding the genetics of longevity as such, and it consists of two groups: offspring of individual with exceptional longevity, that is OPEL, where at least one parent have survived to 95 years or older, and the other group is offspring of individual with usual survivor, where neither parent survived to age 95. So we had like 880 participants with both the proteomics data carried out using the soma scan assay as well as frailty defined. These are the variables which were used for defining frailty, which consist of ADL, the complex diseases, all the cognitive score, gait, grip strength, etc. So almost 41 variables went inside the score, and we could define in 880 participant, and mean frailty Rockwood frailty score was 0.16 plus or minus 0.8. So mainly it's not like that the frailty can go till 1. It get exhausted by when it reaches 0.5 at maximum. Then we have been working with understanding the biological signature of frailty for some time, and using this SOMAScan assay, we found out 143 proteins to be associated with frailty as well as different pathways. But today my focus is on whether can frailty risk be derived from plasma proteome. So here what we have done is like the associated 143 proteins go into the model, and frailty index was regressed on this 143 protein, and using elastic net regression we come up with a 25 protein signature with which we predict frailty index in longevity, as well as we have ARIC cohort as well as Baltimore longitudinal study of aging, that is a BLSA cohort. So we validated in almost like 6,000 participants. So these were those 25 proteins which were selected out from the model, and HTRA1, that is HTRA serine peptidase 1, which was the top positive contributor in the model, while NDST1 was the negative contributor in the model. And also you could see many non-protein like leptin, GDF15, Then immune regulation related interleukin 1RA, troponin T, which is heart muscle indicator of a heart muscle damage in the model. So today I won't be talking about the biology as such, but how this model is playing around. So if you look at the correlation between predicted frailty and the actual frailty index, it's coming to almost 0.6 at the first baseline wave. Then after 1 year, the participant come back to the study, the correlation remain at 0.52, and even after in the third year also, the correlation is pretty high for a complex phenotype like frailty. Then this was in longevity cohort. Then we went to ARIC, where we had almost 5,000 participants. We could replicate the same finding. The correlation was almost 0.6, and the distribution looks almost similar to what we have seen with the frailty. Then we looked how PFI, the proteomic frailty index, was doing with the physical frailty. So this is in the ARIC, where robust participants had a lower PFI compared to pre-frail as well as frail, which was highly significant. So the same thing was observed in ARIC as well as BLSA. Then we looked how the proteomic frailty index was doing with mortality. So this is how the Kaplan-Meier plot looks with the frailty index as such, where individuals with higher frailty are at a higher risk for mortality. But if you look at the PFI which we derived, it's almost looking same. And if you look at the highest tertile, the mortality rate was like 45% of persons succumb to death in with the frailty index, and similarly we could see in case of PFI too. And this association remains significant even after adjustment with the actual frailty index. Then the same thing we could validate in ARIC as well as in BLSA. So also the proteomic frailty model has a good discriminatory ability similar to the actual frailty index. Then the next thing which we looked was how this proteomic frailty index was associated with age-associated phenotype in the longevity in which the model was derived. So we could see like our proteomic frailty index was associated with most of the complex diseases as well as with like higher triglyceride being associated with higher PFI. While you could also see higher PFI was significantly associated with low gait velocity as well as cognition too. The same thing which we could replicate in ARIC, where you could see we had like more cases in ARIC because we had like almost 5,000 participants. And so most of the phenotype which we assessed was associated with higher PFI. So same thing you could see HDL was protective, then triglyceride as well as glucose was risk, and here you could see the grip strength which had like highest association. So as we have seen in the last talk, signifying the importance of grip strength. Also PFI was associated with most of the cognitive score also. Then the similar pattern was observed in the other cohort, the BLSA 2. Then we looked at how PFI is associated with incident dementia. So PFI predict incident dementia too. Then we looked at how which organ system aging is accelerated with frailty. We know that frailty is a multi-system aging syndrome. If it is like aging in a single cell type of organ, we have the chronic diseases. When it's like multiple organ, it's multimorbidity. And if in case of frailty, aging must happen in most of the tissue or organ. So we had like the organ aging clock, which was published in December 2023 in collaboration with Stanford University. So where we could define different organ aging clock as well as like individual who are aging at multiple organs too. So we looked at how this PFI was associated with this PFI was associated with this organ aging. We could find out that most of the organizing measures were indeed associated with PFI. In conclusion, a new protein-based measure, that is PFI, for predicting frailty was derived. PFI was strongly correlated with baseline frailty index as well as longitudinal frailty index, associated with multiple age-associated measure and phenotype, including mortality, and we could validate in diverse cohort. It was derived in Ashkenazi Jewish individual, but it was validated in biracial cohort of ARIC as well as BLSA, and significantly captures cognitive measure and incident dementia associated strongly with biological aging. So might be a new promising measure to capture overall well-being, health, as well as risk of the individual from the blood itself. So it's something different from the aging clock. So I would like to thank Professor Varghese as well as Barsilai, as well as our team at Einstein, as well as our collaborators from NIH as well as Stanford University. Thank you.
Thank you, Dr. Sathyan. That's congratulations on a very comprehensive and research that's, I think, covered all processes of aging, and the fact that we can now plot a subjective feeling like frailty on paper, I think that goes on to show what great strides that we have taken. Any questions from the audience?
Organ aging. We are talking about organ aging.
Yeah.
Is it available for all of us?
We even the proteomic frailty index, if we have like the soma scan assay done, we could compute the proteomic frailty index as well as even the organ aging too. Like it's basically, this is like we should know the expression of the protein as such. So we could compute it, but it's expensive. Soma scan, yeah.
You showed some box plots, and they showed some significant overlap. So the problem with p-values for large cohorts is always that p-values become irrelevant in that regard, that p-values are really driven by the number of samples. So the question is then, when you have for one person, one particular proteomic frailty index value, what's the positive or negative predictive value, specificity, sensitivity? Is it really kind of, again, there's a difference between statistically kind of congregating all the values or having one value for one patient. What information level can you get out of that?
That we have to look into. Like now we could say like higher proteomic frailty index predict multiple things. So actually how much that value decide, like have to be decided.
Thank you.
Yeah.
Can I just add to that?
Yeah, sure.
Yeah, just in terms of scientific validation, can we track the proteomic index and correlate it to clinical symptoms and clinical other parameters that we are already measuring to see if that's valid or not, instead of using just statistical values? Would that be fair to say?
Yeah, we could do if you know the expression.
Correct.
Yeah.
So instead of just using papers as a marker for individual frailty, wouldn't it be more relevant to use already available clinical parameters and correlate it with your parameters?
Yeah, definitely.
Yeah.
Thank you, Dr. Sathyan.